1 LTR.
Well, thank you very much. And I am also thankful to the organizers for providing this opportunity. So today I'm going to talk about the role of CBF1 in HIV latency. So we found that CBF1 induces both the establishment and the maintenance of HIV latency by recruiting polycomb group co-repressor complex at HIV LTR. So as it is well established that HIV latency is a multifactorial phenomenon involving different mechanisms, means various factors and various mechanisms play a role in HIV latency. The two most prominent mechanisms which play a major role in HIV latency are, as it is known from quite a long time, that quiescent T cells lack some important transcription factor such as NF-kappa B and NFAT, which play important role in HIV transcription initiation. And we and Andy Rice has demonstrated that quiescent T cells also have reduced level of PTFP, and PTFP is essential for HIV transcription elongation. So these two major mechanisms describe HIV latency due to defect in either initiation or the elongation phase of transcription due to lack of some important transcription factor in quiescent T cells, and therefore virus is unable to do its transcription and become transcriptionally silent and become latent. But the same outcome can also be achieved if the quiescent T cells have a higher level of transcription repressors than the activated T cells. So the question we ask is, are there specific repressors of HIV LTR promoter which are higher in resting T cell than activated T cell counterpart? So after a long search, we come up with an candidate factor which is a strong repressor of HIV transcription, and also it presents predominantly in the resting T cells, and its level drops sharply following T cell activation. The factor is known as CBF1, it stands for latency C promoter binding factor 1. And so to demonstrate the kinetics of CBF1 in resting and activated T cell, we activated the T cells via TCR stimulation by anti-CD3, CD28 antibodies treatment. And we analyzed the mRNA level of CBF1 at different time intervals of activation. So you can see before activation, the cell has very high level of CBF1. However, after activation, cell activation, the level of CBF1 drops sharply and remain low for at least 3 hours, the time period we analyze the level of, we perform these experiments. But in contrast to the CBF1 level, when we analyze the HIV mRNA level, which represent the ongoing HIV transcription, then after TCR activation or T cell activation, the level of HIV transcription went up, demonstrated by the high level of HIV mRNA. So these results demonstrate that there is an inverse correlation between the cellular level of CBF1 and HIV transcription. So, and also suggest, that maybe CBF1 plays some important role in HIV transcription, as when the cellular level of CBF1 are high, the provirus is latent. And after T cell activation, when the level of CBF1 drops, the latent provirus get reactivated. And in fact, in our published studies, we have confirmed the important role of CBF1 in HIV latency, not only in transformed T cell lines, but also in primary T cells, by developing a model system for HIV latency in primary T cells. So briefly, we have demonstrated the direct role of CBF1 in HIV latency, as when we knock down the CBF1, endogenous CBF1, in latently infected cells, the latent proviruses get reactivated. And how it does, and that is CBF inhibit HIV transcription by recruiting HDAC containing co-repressor complexes. And those HDAC histone deacetylases then deacetylates the core histone and induce transcriptionally repressive chromatin structure and inhibit transcription. So these results demonstrate that CBF1 induce HIV latency by recruiting co-repressor complexes at HIV LTR. So to extend these studies further, we wanted to know what other repressive epigenetic modifications are induced by CBF1 in order to restrict the transcription of HIV during HIV latency. So we performed some chip assay, chromatin immunoprecipitation assay, before and after knocking down the endogenous CBF1 protein in latently infected Jurkat T cells. And we analyzed the corresponding loss of various epigenetic marks and epigenetic factors at HIV LTR due to following the knockdown of CBF1. So you can see when we knock down the CBF1, the red bars, the latent provirus got reactivated, demonstrated by the high level of recruitment of RNA polymerase II, so representing the ongoing high level of HIV transcription. But interestingly, when we knocked down the CBF1, we found the corresponding loss of two repressive epigenetic modification, or you can say heterochromatic marks, namely histone 3 methylated at position 27 and histone 3 trimethylated at position 9. And these heterochromatic marks or heterochromatic modification are known to induce transcriptionally repressive chromatin structure. And the importance of these heterochromatic marks and modification has already been established in HIV latency by several groups, including us. Especially, we have demonstrated the importance of these heterochromatic marks in establishing HIV latency in primary T cells. So these results demonstrate that CBF1, in addition to the histone deacetylation, also induce the repressive histone methylation in order to restrict HIV transcription during HIV latency. And these results also suggest that CBF1 recruited co-repressor complexes carry the enzymes which induce these heterochromatic modification. And in fact, we found the corresponding loss of EZH2 following CBF1 knockdown. And EZH2 is an enzyme which catalyzes the methylation of histone 3 at position 27. And the importance of EZH2 in HIV latency has recently been demonstrated by Jonathan Kahn group. So to extend these studies or these findings further, here we demonstrate that CBF1 is the protein which recruits EZH2 at HIV LTR. And interestingly, EZH2 is also a core component of polycomb group co-repressor complex. So it suggests that CBF1 induces HIV latency by recruiting polycomb group co-repressor complex. And polycomb group co-repressor complexes are known to repress the transcription of cellular gene by inducing multiple layers of repressive epigenetic modification. Means that when they repress the transcription, they repress by inducing various epigenetic modification, not just one, but they induce simultaneously various epigenetic modification. And therefore, polycomb group co-repressor complexes not only play a role in the transient silencing of the genes, but also permanent silencing of cellular genes, such as, for example, the permanent silencing of endogenous retroviruses. So to confirm that the CBF1 induces HIV latency by recruiting polycomb group co-repressor complex at HIV LTR, we analyze the corresponding loss of various core components of polycomb group co-repressor complex at HIV LTR following CBF1 knockdown. So you can see, so these are the core components of the polycomb group co-repressor complex. So when we knock down the CBF1, the level of these components went down at HIV LTR. Means when we remove the CBF1, these factors also gone down. So demonstrating that the CBF1 is responsible for their recruitment at HIV LTR. So these results demonstrate that CBF1 induces HIV latency by recruiting polycomb group co-repressor complex at HIV LTR. And interestingly, the JARID1A is a histone demethylase, which removes the methyl group of histone 3 at position 4. So in contrast to the methylation of histone 3 at position 9 and 27, which I demonstrated to you earlier, which are the heterochromatic marks, induce transcriptionally repressive chromatin structure and inhibit transcription, the methylation of histone 3 at position 4 is a euchromatic mark, which establish transcriptionally active chromatin structure and facilitate transcription. So JARID1A is an enzyme which removes that euchromatic mark. So what it demonstrates that polycomb group co-repressor complex not only contain the enzymes which induce repressive chromatin structure, but also contain the enzyme which remove the active chromatin structure. So what it suggests that CBF1 by recruiting the polycomb group co-repressor complex at HIV LTR not only facilitate the establishment of HIV latency, but also support the maintenance of HIV latency by recruiting polycomb group co-repressor complex. So next, we wanted to confirm the direct role of polycomb group co-repressor complex in HIV latency. So for that, we performed some knockdown experiment where we knocked down the various core component of polycomb group co-repressor complex in latently infected Jurkat T cells, and we analyze the corresponding activation of latent provirus along with azadeoxycytidine, inhibitor of DNA methylation. So you can see when we knock down the various core component of polycomb group co-repressor complex, you can see twofold more reactivation of latent provirus. So these suggest that when we remove the restriction posed by polycomb group co-repressor complex, the latent provirus got reactivated. So these results clearly demonstrate the direct role of polycomb group co-repressor complex in HIV latency. And overall, these results demonstrate that CBF1, by recruiting the polycomb group co-repressor complex, facilitate both the establishment and the maintenance of HIV latency. So to summarize, so we demonstrate that earlier, the CBF1 is a potent and specific inhibitor of HIV transcription, and CBF1 performs its function by recruiting polycomb group co-repressor complex at HIV LTR, and like an ideal repressor, CBF1 level goes down following T cell activation, and multiple chromatin modifying mechanisms are involved in regulating HIV gene expression from latent provirus. And interestingly, the inhibitors of polycomb group co-repressor complex may play a useful role for the induction strategies designed to eradicate latent viral pools in HIV patients. So in other words, instead of targeting individual epigenetic modification in order to reactivate latent provirus in our shock and kill strategy to eradicate HIV virus, we can target the complexes, for example, the polycomb group co-repressor complex. And as I mentioned, it induces various repressive epigenetic modification in order to repress gene. And so by targeting the complex, we can simultaneously target the various repressive epigenetic modification, and that could result in a better reactivation of the latent provirus. And also, these study also suggest or establish another target of the therapeutical importance, target of therapeutic importance in the form of polycomb group co-repressor complex. And I like to thank Professor Jonathan Carr, because some of these experiments were performed when I was in his lab. And I am also very, very thankful to the MFAR grant for supporting this study and several other studies. And I'm also thankful to the CFAR grant from Case Western Reserve University. And thank you very much for your kind attention. And if you have any question, please go ahead.